The issue of chemicals in the environment is a story about science, politics, and humanity's relationship with the planet. My name is Chris Hughes, independent scientist, and this is the Chemical Journeys podcast. Join me as we uncover the story of chemicals in the environment through conversations with individuals working in this space. So, let's get started. Hello everyone, and welcome to the Chemical Journeys podcast. Today I'm speaking with Professor Thomas Hartung. Thomas is a professor of evidence-based toxicology at Johns Hopkins Bloomberg School of Public Health and of pharmacology and toxicology at the University of Konstanz. He's also the director of the Center for Alternatives to Animal Testing and the field chief editor of the journal Frontiers in AI. Thomas, thanks for joining me.
Yeah, thanks for having me.
I'm really excited to speak with you today, Thomas. It's a little bit coincidental that I came across some of your work. I saw one of your papers on how AI can deliver the human exposome project in Nature Medicine. And I have to say, it was a fascinating read, really visionary. And I also get a strong sense of optimism from the work that you're doing and excitement about everything that's happening. I myself am not so involved in the NAM space, new approach methodologies, but given the work that you're doing and the perspectives, I'm very happy to talk to you today. So perhaps we could start, if you like, provide us a bit of information about your background and your research interests in your own words.
Yeah. Okay, there's two types of researchers. Some choose a topic and dive deeper and deeper and become popes in their field. I'm the opposite. I'm broad. I'm doing lots of different things in many areas with many affiliations because I like to connect people. I like to bring different areas together. And out of my work into mechanisms of medicine and drug development and some toxicity, my topic found me. I developed some alternative methods. First alternative to the rabbit pyrogen test, which now 30 years later are saving 150,000 rabbits per year in Europe alone. This made me take a job with the European Commission to head their validation body, the European Center for Validation of Alternative Methods. And this is where I considerably had to broaden my spectrum, also going for biologicals and vaccines and ecotoxicology and computational methodologies, because I was suddenly responsible for all of these. But ultimately, I started to understand that the animal tests which we are using are terribly outdated, and that we have at the same time more and more new methodologies, transformative technologies, which promise to do better.
Thank you for that. So a great introduction. And you've recently described that animal-free science is going to become like what the automobile was to the horse in the early 20th century. Could you tell us what you mean by that?
Yeah, we see that there is a transition between technologies, an overdue transition. The animal models in toxicology, where I'm doing most of my work, were introduced when I was not yet born or in kindergarten, and I'm turning 62 now. So what is really important to understand that this is also a normal thing, that we get something new. Our knowledge in the sciences is doubling every 3 years, and this alone means there's a lot in the box since we developed these animal tests. Very similarly, we had a technology away from animals 110 years ago, roughly, when cars came. And what I really found interesting, digging a bit deeper, how much of the argument was just the same. Yeah, how reliable these horses are and how much experience we have with them and these dangerous technologies. And what sounds now laughable when you hear how people were arguing we will never replace the horse is something I see coming forward we're doing. And people will in doubt laugh about us and about the hesitance in really doing something modern and timely.
Yeah, I think as humans, we are generally quite resistant to change in many aspects, right?
Yeah, especially in the safety sciences. Yeah, there's a good reason. I don't argue for going for the latest fashionable thing. We need to establish safety. We have a big responsibility in doing so. But at the same time, we need to adapt to scientific progress. And what we have been doing is, in toxicology in particular, we did create a quilt every scandal gave a safety patch and one patch on the other. They're all of their different making and sizes and material. And altogether, this gave a really very rigid system where you occasionally replace one patch with an alternative method, but you don't really change the overall construct. This is the type of discussion I'm trying to have. How much can new transformative technologies allow us to make an actual change?
Yes. And maybe you could just explain a bit about what are these technologies that we're talking about?
I mean, there's in essence, quite a few. You could take technologies like omics and sensors, as the type of things which generate much more data. I mean, toxicology has been a data poor field. Most of the things you could do with a handheld calculator, counting animals or the number of tumors in an organ. But it is becoming a big data science now. And this leads me to the first of the really transformative technologies, which is the artificial intelligence movement. I mean, toxicology is no different to all of the other areas of our life. I believe that soon we will have to write science instead of IE with AI, because science is changing because of this. The other side is the bioengineering has allowed us to really move cell culture to something much more meaningful. So instead of using some tumor lines and keeping them in petri dishes alive and measuring whether we can kill them, we are coming more and more to systems which are recreating the architecture and functionality of an organ. So these are necessarily co-cultures. Take a human lung, these are more than 40 cell types. And its function is not to have the cells survive, but to breathe. And you can make similar arguments for each and every organ. And it is mainly the advent of stem cell technologies which gives us access to high quality human cells.
Yes.
You get skin, perhaps, get blood quite well, but otherwise it is very difficult to get in higher quantities and reproducibly any human tissues. I'm working on the human brain. If I want to pick your brain and mean it, you will not be very forthcoming, I see. And this has changed really dramatically, first with human embryonic stem cells, but then ethically much cleaner 7 years later with the induced pluripotent stem cells, which also have the dramatic advantage that you know who the donor is. Plus, in vitro fertilized embryos, you don't know what they would have become, what disease history they would develop. The organoids and similar we can do now from a patient donor, they have a disease history, whether this is a person prone to allergies, whether they developed Alzheimer or whatever type of disease. And very often this is reflected in the cell models we can generate from their cells.
You know, it does sound absolutely amazing what we're doing now with this technology. It's really getting into the realms of science fiction.
Oh, absolutely.
Maybe you could compare that to the animal models. I mean, tell me, what are the problems with animal models?
You know, when I took the job to promote and validate alternatives to animal testing, my understanding was, okay, this is motivated by ethics and by the fact that a broad part of society wants us to move out of animal testing. About 60% of Europeans and about 50% of Americans are objecting to medical testing on animals. And that's quite a number, and policymakers listen to this. But the longer I'm in the field, the more I understand that is also economical reasons and scientific reasons to change. I said once, very simply, we are not 70 kilogram rats. And there's a lot of truth in this. And also modern drug development is now developing largely biologicals, no longer these small chemicals, which you can still argue whether the animals are representative. They're certainly not representative for human proteins or antibodies against human proteins. And most animal testing is useless. 57% of the new drugs are such biologicals. And so pharma needs it. And the regulators in the FDA and EMA need them to really judge these systems, because even non-human primates don't do a proper job in most of the cases. The other side is that animal tests are costly and slow. This does not fit modern development cycles for products. If you take an example, cancer is certainly the thing most people would like to know if it's about safety of chemicals. we hardly test any substance for cancer, because the only test we have accepted is an animal test, the cancer bioassay. And the cancer bioassay takes you 4 to 5 years to get a result, because you have to treat 400 rats for 2 years of their life every day. And then it takes you about 2 years to analyze all of the organs and samples and slice all of these with preparation and then reporting. You're at 4 or 5 years easily. Yeah? And then what do you get in return for an exercise which does cost you almost 1 million? Yeah? You get more than 50% positives, because these animals at the end of their 2-year lifetime, they are full of cancer anyway. So there's a lot of false positives in these, many more than actual estimates by experts would suggest. So it is very difficult for companies to accept this, but even regulators don't accept the results. An analysis has shown that 58% of the positive cancer bioassays did not lead to a classification by the US EPA, because they said, no, it's not relevant. Yeah, and we really have sometimes absurd situations of having to handle then false positive results by these cancer bioassays. And another thing which people often overlook, in order to feed 400 or inject 400 rats for 2 years of their lifetime, you need about 10 kilograms of the substance. Tell a chemist to produce 10 kilograms of a chemical which is not yet in production. It is essentially impossible. So there is a lot of value proposition of getting some data, and they don't even need to outperform the cancer assay, but just to get something which says, oh, there's an alert, we should pause and look into this. You get to get it fast on a small quantity of things, and that's exactly the value proposition of these in vitro systems.
Yes, I mean, it sounds like there was huge scope for refining the way that we've been doing this for many decades. Another question I have about these new methods, so you alluded to some of the challenges to their adoption. Maybe we can talk about some of those in a bit more detail. I think you mentioned validation being one aspect, so presumably this is about demonstrating that the tests are robust and relevant.
Exactly. I had this job for 7 years in Europe to validate these methods, because you cannot have every time a company is coming with a new test to a regulator, need them to argue for their test and the regulators to find out, is it trustable, is it not? Yeah. So the idea of simply saying there should be an institution which follows certain guidance in order to assess, and then come to the result, yeah, can recommend this assay or not. And then both sides can talk much easier to each other and can expect that also the next regulator in the next country will have the same information basis, because it's in the meantime a very international exercise, standardized through the OECD. So that's the basic idea. And some 50, 60 tests have been internationally validated, made their way into the OECD test guideline program or pharmacopoeias in the world. So there's a substantial number. But we also have to say there's still a lot of animal tests where we don't have an alternative officially recognized.
Yes.
And the two things you really want, or actually you want to do 3 things according to the OECD guidance. The first thing is you have to show it's reproducible. Yeah, you get the same in whatever lab is doing it.
Yeah.
The second thing is you want to show that it is giving you relevant results. And this has been for many years now been interpreted, it is giving the same results as the animal test did, which has a big inherent problem, which is if the animal test is not perfect, how do you reproduce something imperfect? How do you get out of the shortcomings of something which is not reflecting humans, for example? Yes. And the third element is, and this is something which is a principle, but is not really executed very much, is it should be scientifically sound. So it should reflect ultimately our scientific understanding of a certain health effect of a disease.
Yes.
And this is something which I think is actually the big potential, because I call this mechanistic validation.
Yes.
I don't care whether we can reproduce a finding in an animal. If you take thalidomide,
Yeah.
thalidomide is not showing exactly in the animals we are using in rats and mice. For this reason, I don't want to reproduce that thalidomide is negative. I want to show it positive. Yeah.
Yeah.
And this is really very important. But I also want it to be positive for the right reason. I want to be positive because it is triggering a relevant mechanism for humans. Not it is a completely different mechanism leading to a completely different toxicity for a given chemical.
Maybe on that point, because I was thinking about this, you have all these different assays, in vitro assays, and microphysiological systems that don't. It's kind of like a constellation of tools, right? That's designed to inform a particular question about an adverse effect of a chemical, whereas before you may have had one animal test. A big question that comes up also in the area that I work is, how do you know that what you're seeing in the test is going to be relevant to either an individual or on a population level? And so I was thinking with these assays, and I think you've alluded to this, that we have some mechanistic understanding that we used to develop a hypothesis that we then seek to observe from the tool, but how much of the validation is coming kind of from the bottom up as well, in that we need to generate data with these tools in order to connect this, to validate our mechanisms, our understandings?
Yeah. I mean, actually, it is, if you give it a closer look, we are very poorly using mechanism in our validation studies. I have often called for the need for mechanistic validation, putting this out. And there's a very interesting movement with the adverse outcome pathways really to increasingly agree on mechanism. But we are only starting to translate this also into our validation procedures. But I think that's where really a lot of progress will lie. The other thing is, us toxicologists, we like it black and white.
Yeah.
So we want one test, and this one test is classifying chemical. And there is a tremendous flaw to this. First of all, no single test can deliver and give us all of the different answers. Yeah? For example, a test can only be either sensitive or specific. If I make them very sensitive, I pick up all of the toxicants, but then I also have quite a few false positives, like in the cancer assay. Or I make them highly specific and can trust, but then I miss a lot.
Yes.
And this is already a very big problem. So it is often much better to have a screening test and then a confirmatory test to follow. And this already requires a combination of assays. And then black and white get a lot of gray.
Yes.
But our classification needs always meant we need a definitive test,
Yes.
the one which is the gold standard. So I really believe by moving away and also accepting that we are dealing with probabilities, I like to call it probabilistic risk assessment, I think we do a favor to science and what we can really deliver. And also, any test is contributing to identify the degree of gray and is helping us to reduce the uncertainty around this color gray.
Yes.
But none is a game changer. And we can have much less fear about the new methods when they are only modifying an assessment. They're not making something which was black before, now white, or the other way around, which is always the big concern.
Yes.
And interestingly, current technologies, especially artificial intelligence, is doing exactly this because it is a highly probabilistic approach.
Yes. Yeah, of course. I see a lot of similarities here, actually, in the area that I'm working in, which is mainly focused around environmental fate and biodegradation assessment. It's the same problem, right? The tests are very expensive. You take one shot and then you have to make regulatory decisions out of that. And although it's written into the regulation that you should employ what's called a weight of evidence, so you should try to work with all the evidence that's available to come to a single conclusion that weighs all of that evidence. But the reality in terms of how things play out often falls into the black and white, like you say, of, looking towards a worst case result. On that point as well, you mentioned reproducibility. That's another big challenge with biodegradation testing. And I wondered, because you mentioned that you have donor cells for a lot of these assays, how important is the genetic component towards the reproducibility of results?
Incredibly important, because we are just not the same. Yeah? And we have, by running our experiments on strains of mice or rats, which are genetically very limited, whether they're inbred or outbred. Yeah, in one case, being identical twins, even. Yeah, this is reducing what is normally making us different, and we see. This is why we can get some results with groups of 10, 20, 40 animals, while for any clinical trial for the same substances, you need thousands of people to find out, because we are so different. Mm-hmm. And all these attempts towards personalized medicine, some people are also talking about personalized toxicology, this requires us to recognize what makes us different. And this is not only genetics, it is also disease history, age, gender, obesity, so body weight and other factors which we have to recognize. Highly standardized rats, 3 months old, are not reflecting any of this. Also, you can say in 40 years of a human genome project, we have learned that only 5% of diseases are purely genetic, and about 40% have a genetic component which we could identify. But there is a lot which comes really from exposure. And the exposure is even more varied and more dynamic than the genetics which we are showing. So I think there's a lot to be understood here. And this is why we're arguing, for example, for a human exposome project.
Yes. So is the approach to use a fresh donor stem cells each time you do these assays, or is there a cloning process going on?
I mean, at the moment, it is already something that we ask for several donors in most of the experiments. That's becoming more or less a standard for stem cell derived tests, and there's not yet any guidance on how many you actually need in order to be comprehensive. But the National Center for Advancing Translational Sciences has already started to do clinical trials on a chip. So this is a program they are running where they really want different patients reflected in stem cell models and see whether their stem cells would have predicted the individual's outcome in the clinical study. So we're getting to something which is more personalized and is helping us also to see distribution over patient populations or healthy populations.
Yes, it's really fascinating, and that just highlights how we need to rethink our methodologies and our processes now that we have the opportunity to generate larger volumes of data and we can approach things in a different way. I think you mentioned education being another important component of this. So do you want to say any words around that?
Education is critical, of a general population. It is quite amazing that people 500 years after Paracelsus have not yet understood that the dose makes the poison and are permanently arguing about the presence of substances, but also the education of the next generation or of the practitioners of today. We see that a lot of the resistance in accepting new methodologies comes from the people who have never been trained on them, especially in something which is moving as fast as artificial intelligence is doing now. This means an enormous hurdle. I think everybody understands there's something in the box and we will be outdated if we are not embracing it, but how to learn it, how to decide what is hype and snake oil salt to us, and what is just delivering makes it very difficult. Some people are validating, assessing the things, and are building the trust for others who don't have the means and the time to evaluate all of these tools.
Yes. Yeah, and trust is a big component there, isn't it? And I suppose the general problem in society is degrading trust in institutions and science in general. Are you coming up against those kinds of challenges?
Yeah, I mean, I have to say, first of all, we are dealing with the trust of the practitioners in our field, and these are scientists, and scientists in general trust in science.
Yeah.
So we don't have the same problem as with the general public, where we see that social media are eroding what we can get across. But I also have seen often that people, if you approach them with a risk communication, which is tailored to their needs, you can achieve quite a bit of understanding and interest, especially if you start engaging them into some type of dialogue. All of these things are now augmented also with these AI tools. These chatbots essentially feel like talking to a human expert, but they can scale the interactions of the experts with the general public and make it much more intense, how it is tailored to the specific needs. There's really revolutionary changes in how we can produce educational measures and provide information which is sourcing broadly, so to get you much better information than you could possibly get in the past. And at the same time is packaging it to your needs and getting you where you are and giving you the information in a way that you ultimately can grasp it.
Yes. Yeah, I think AI is just a general theme throughout this conversation. I did want to just touch on, there was a pretty big announcement recently with the National Institutes of Health in relation to these methods.
Yeah, I mean, we see at the moment in the US a dramatic change in the landscape, which came actually first from the two regulatory agencies. The EPA already in 2019 announced they want to move out of animal testing as a standard requirement by 2035, which was a very, very surprising and strong statement. And this pledge has been renewed in April this year by the new head of EPA, Zeldin. So the EPA is still on track for doing this. And the FDA, which is arguably the most important regulatory agency in the world, they announced the roadmap. They don't want a roadmap. They announced the roadmap, which said we want to reduce animal testing and we will start with monoclonal antibodies and then everything else follows. Pretty strong, again, timelines for this. I say they're the most important regulatory agency. It's very simple. Most of the changes and developments take place in pharma. And the pharma market in the US is second to none. We are only 4% of the world population, but we are 53% of the drug market of the world, 67% of the drug market for the new drugs under patent. Yeah. So every pharma company in the world is just looking what FDA wants. Many actually don't even register their drugs in Europe and other parts of the world because they're just fearing they could later be told, why can you sell so much cheaper here and not in the US? So FDA's announcement was spectacularly important because it's a signal to their own people. So within the agency, I mean, this is a 19,000 people agency. Yeah? They have tons of people that all need to understand what is the new direction. Yeah? But it is also signaling that the FDA Modernization Act, which was asking this from the FDA 2 years earlier, is actually being translated into action now. It is a signal to the regulated industry, as well as to biotech companies, that there is a market. Yeah, there's something to gain which will accelerate progress. Only some 14 days later, the NIH announced they want to refocus their funding on human relevant systems. They want to stress more, especially these complex in vitro systems, microphysiological systems. They are naming these. And they even came out now with a statement saying they will no longer publish any calls where they only request animal studies. They want as a regular thing to call also for the alternative and new approach methods. And I think this is really an important signal again for the scientific community. We have to work together. Yeah? It is what I called sometimes in vivitrosi. So it's in vivo, in vitro, and in silico combined. And these old-fashioned approaches of I do my animal experiment and I don't care about human relevance will no longer fly with the NIH. Sure, they leave it as usual to the reviewers then to implement this. And if the reviewers select pure animal studies, I think they will not stop funding these. But the request, first of all, and the call texts they were publishing will be stressing very clearly, we would like to see the human relevance embedded into any type of study.
Do you see this as a major lever that will bring to light more the disparity between the animal models and these technologies, or do you think that we're already clear on that?
I think it is absolutely obvious for me that this is going to happen. We see that different animal species in toxicology, where we sometimes do these things, reproductive tox on rabbits and rats, or the cancer assays on mouse and rats, we see that they predict each other by about 60%. And there's no reason to assume that they predict humans any better. On the contrary, I would expect that this is going to fuel this discussion of transitioning to human relevant system for human research.
Yes.
And I think this is helping us to finally have this discussion, which was very much avoided by the animal experiment community. They have resisted the formal validation of their assays. We don't have a lot of assays which were really undergoing ring trials to show their reproducibility, if not even their relevance. We have really learned the hard way that very big drug programs have likely failed because of misleading animal results.
Yes.
It is the lack of relevance of the Alzheimer animal models, for example, that we have spent more than 150 billion in drug development for Alzheimer without really remarkable progress in drugs which are helping here. Yeah?
Wow. I mean, that really brings it to light when you think about what we've lost as a society by not taking action with some of these things as well. Yeah.
But we should also be fair. The cell culture is around in a reasonably infrastructured and reliable quality of products since the 80s, 90s, not much earlier.
Yes.
In the 80s, when I started cell culture in a lab, a lot of reagents were still done at home.
Yeah.
And computers are only most recently powerful enough to really make these large predictions. And it is really the advent of AI.
Yes.
But for example, to design a drug, AI is powerful enough, and this came with the power of computers, not more ingenious programs, since about 2019. So 2019 is the starting point for most of the projects which designed drugs by AI. We call them AI first drugs. In 2023, already 18 AI first drugs made it into human trials. That's a remarkably fast preclinical development and shows also the value proposition of these tools. Yeah? In 2024, the first of these 18 already completed a successful phase 2A in patients and went into a phase 2B study, for the one by in silico medicine for lung fibrosis. And this is unheard of. They started in 2019. In just 5 years into clinical phase 2, 1 day earlier to the market is worth for a pharmaceutical company about $1 million.
Wow. So you can really see the potential upside with embracing these technologies as well. You've already mentioned about how AI sort of accelerates the distribution of communication. So we have the communication flows that should enable these things to be adopted rapidly. We don't want the human resistance sort of biases, cognitive fallacies, or, presuppositions to be another bottleneck. And I guess the other one is on the regulatory side. And you've already mentioned that there's these big movements in the US. What's happening on the EU side?
I mean, the EU is much longer committed to change. It's quite remarkable that already in 1986, the European laboratory animal welfare legislation said, whenever there is an alternative reasonably available, you must use it. This is very strong. And they also committed the Commission and the member states to further these developments. This became even stronger with the 2010 revision of the legislation. So traditionally, Europe has been at the front, but it has been very much driven by animal welfare considerations, so public opinion, and slow. I think the discussion about a roadmap to phase out animal tests, so something similar what the US just announced, has been ongoing now for 6 years. And there's a plan now to announce a draft of this roadmap in the beginning of 26. Yeah? And you see, it is moving, and it has a broad basis, but sometimes it is also ridiculously slow.
Yes.
Yeah, and I really commend the US here by just saying, yeah, let's do it. That's an engineering exercise. We want technologies which do the trick. And we have been convinced that the new stuff can do it. And we see the shortcomings of what we have been doing in the past. So let's give it a shot.
Yes.
And they don't say we just blindly buy it. They say just want to now enable such a transition.
Yeah, and they can see there's advantages there in many aspects, health improvements, but also market advantages as well.
Yeah, absolutely. No, this is an industry. I was very surprised to see data from Markets and Markets, which is a market research company, You can argue their numbers and their approach, but they estimated the in vivo toxicology market at about 4 billion, but they estimated the in vitro market right now at 14 billion. So the companies are really doing a lot of this already, while it is not as prominent because it's used for internal decision taking. They don't use it regularly for registrations because they just follow a checkbox approach.
Yeah.
And they would be stupid to offer more data than they asked for. But a lot of the decisions internally are based on other type of systems. Yeah. And this is why, compared to the 70s, animal use numbers in Europe or the US have gone down by 2/3. Yeah, so we had much more animal use in the 70s, where pharma was the dominating user, and they have dramatically reduced animal testing by such other search strategies. Yeah.
Yes. When I've heard discussions about this on the regulatory side in the EU, it's always couched in those animal methods, and we have, for example, in the REACH regulation, we have a standard set of information requirements and you need to tick all the boxes, and that has sort of framed everything. And from what I hear, it's very difficult to change that aspect of the regulation. So, but can you see that moving in the near future?
Yeah, and my personal career has been very closely linked to this. I joined the European Commission in 2002. And at the time, they were handling the first draft of the REACH legislation. And at the time, it did not even mention alternative methods. And I pride myself that in the end, when it was decided in 2006, the promotion of alternative methods was one of the 3 goals of REACH. Most people in the regulatory area seem not to read the full Article 1. Yeah. They stop after the first 2, but that is a bit cynical. Yeah. But I was also then responsible for leading 200 experts to write the guidance for industry, how to implement REACH. And we took a lot of effort to create some openings for the alternative methods which were not yet available, but which at some point could make it.
Yes.
But I also was a big critic of this enormous use of animals. And we just did 2 years ago an actual count. We counted how many animals have already been used for REACH. And you have to see that the commissioner responsible at the time, Günther Verheugen, he had at some point stated that REACH will not be acceptable if it is using more than 1 million animals. Yeah, we counted now already 4.2 million, which had been used. And a lot of question marks still, which could add several millions to this game.
Yes.
So REACH is an enormous consumer of animals.
Yes.
And again, I would say if we had mechanisms to faster learn from the animal experiments we are doing, we could probably have prioritized better. We could have come up with methodologies and guidance which would be dramatically reducing these animals.
Yes.
But REACH is very much a checkbox approach.
Yes. Yes.
This tonnage level, you do this and this and this. That's it.
Yeah, and I see that also as the regulation is implemented and you see new guidance evolving, and recently Annex 11 of REACH was updated as well around the rules for adaptations, and you see sort of written into it a highly kind of precautionary approach such that you need to meet a very, very high bar to do anything other than the standard animal test. Or another example is with read across, to use the data from one substance to support another substance, there's a very high bar for justifying that use of read across. And there have been some publications recently that suggest that a lot of read across arguments are being rejected.
Yeah. No, I mean, this is, there's this couple of problems which are accumulating. The first one is the staffing and the amount of expertise in the agencies. If you compare that, the US EPA, at least before now all this slashing of posts, was 14,000 people. 14,000, the EPA. And compare this to, I don't know, 700, perhaps 800 now in ECHA. That's a tremendous difference.
Yes.
Yeah? And there's no intramural research. There's not really the possibility to do much more than just following the dossiers and taking an approach. The second thing, policy or legislation in the US and Europe is very different.
Yes.
In Europe, we have legislation which is extremely prescriptive, which says at this tonnage level, do this animal experiment. Yeah.
Yeah.
And it was, at least that's how ECHA perceived it in the past, interpreted in the past, for them very difficult to change these requirements when there was any type of progress. I read the legislation as something which is inviting to exactly do this, because that's what policymakers wanted. They put in a lot of requests for animal testing only being the last resort. But for example, when the extended one generation study for reproductive toxicity testing was validated, then it was extremely difficult for ECHA to accept this to replace the two generation study. Though it brings down the number of animals from about 3,000 to something around 1,400. So there's a per chemical. This is really a very, very big thing, but it took years of arguments to then broadly accept this. And one of the main reasons was, the extended one generation study is not mentioned in the legislative text. How can we then accept it? And I think here the comfort zone of the regulators who are doing this as a first in the world. Nobody has such a legislation. And I'm for public health, I think this is a very, very important legislation. But we also have to learn on the road and have to say, sure, it's not perfect. It can't be perfect.
Yes.
It was done by policymakers, and it was done on the basis of science of 20 years ago. So let's occasionally revise it, simply give the agency, not the policymakers, the opportunity to adapt to scientific progress.
Yes.
And this is better in the US, There the policymakers say, bring information on reproductive toxicity, but they don't specify which test. They would leave it every time to the agency to advise on what is the current state of the art.
Yeah, that's really striking that you mentioned that the number of staff at EPA versus ECHA. I mean, it's 20-fold difference. That really jumps out to me. Other guests I've had on the podcast have talked about the way we've built REACH. It's really a very big, large machine that's designed to run almost... it's designed to be maintained and managed as it goes through chemicals and evaluates them with perhaps not that much reactive intervention.
I can tell you that the main author of the REACH legislation was a mathematician by training. And in some aspects you smell it. I did not know about any other trained toxicologist in the European Commission when I was working there. There had been occasionally at the European Chemical Bureau, but most of the people did come in not as toxicologists, but as biologists and others. And it also took them a while to attract to ECHA in Helsinki more seasoned toxicologists and practitioners. I think it takes long until an agency matures and has also the self-confidence in moving forward. But I see recently very positive developments. In both much more even in EFSA. But there was just one of the keynote speakers at EMA's 30th anniversary, and I have to say I'm impressed how all of these agencies are now handling new approach methodologies more favorably and more progressively, and have set up mechanisms which are not reflecting what is taking place at the moment so dramatically fast in the US, but they're going in the right direction.
Yes. No, that's really positive. It's great to hear your perspective as somebody who's very close to all these developments that are happening in various regions around the world. We've covered many different areas where AI is really revolutionizing things. I think something that would be really interesting for listeners, scientists who are hearing lots about AI but perhaps haven't fully embraced it themselves, would be great to hear from yourself in terms of how are you utilizing AI in your day-to-day work, and what do you recommend are ways for people to get over that initial barrier?
I strongly believe that AI is changing all of science at the moment. I do a lot of talks entitled AI will only replace toxicologists who don't use it, and these type of things. Because of my involvement with Frontiers in AI as a field chief editor, where we have published 1,400 articles in the last 7 years, I see a lot of things happening. I became, for example, part of the World Economic Forum's committee for 10 top technologies. And for them, I wrote with some co-authors last year the white paper on how large language models are changing science. And it's really interesting to change this perspective, what it makes. And I think that actually we are at the moment in the process where the majority of colleagues is just understanding how transformative this is, and that they have to use these tools. So it's a phase of making AI, integrating AI into science.
Mm-hmm.
But I actually believe that we will soon enter a phase where we do science for AI, not AI for science, because it is crazy. But the integration of knowledge and
Yes.
the availability of knowledge, which AI is enabling, is such a transformative step. If you see that in the life sciences, 3.4 million papers are published at the moment every year. 3.4 million. Yeah? Nobody can read these. Yeah.
Yeah.
First of all, because a large part is still hinter paywalls. But
Yeah.
it is estimated that now about 50% of scientific publications are open access, but only 20% are true open access, gold open access in journals which are fully open access, while the others are in repositories and difficult to access and find for AI. Yeah? So essentially, we're working with 20% of the scientific knowledge when training the large language models. But in the future, where we work to AI, we need to understand that everything we publish, which is not open access, which is not machine readable, will not be part of the common knowledge. And
Yes.
AI already for 2 years is outperforming humans in annotating scientific papers. And since then has also solved the problem of being able to read figures and diagrams and tables, which was a problem 2 years ago. So, and while my PhD students perhaps read a paper per week, or hopefully 2 or 3, AI can read millions of paper in a day and will never forget.
Yes.
And is also able to see patterns or just to retrieve the information. Yeah, that's such a value proposition.
Yes.
I was almost 2 years ago doing a peer review for the EPA, where they had a chemical for which 2 people worked 1.5 years to find all of the information on this chemical and then do a little bit of read across to some similar chemicals. 2 people,
Yeah.
1.5 years. That's 300,000 in investment for the
Yes.
EPA, I assume. I found all of the information in 1 hour using AI, and even found an entire risk assessment on this substance, which was missed by the colleagues. You imagine what this is as a value proposition for any type of risk assessment.
Indeed.
And then AI can find patterns, can integrate this information.
Yes.
Already in 2018, we were able to outperform the reproducibility of the 9 most used toxicological tests, which is on average 81%. Predicting for 190,000 chemicals the classifications they had received, we were 87% correct. This is 2018. You have to imagine that AI since 2010, when deep learning was introduced, is doubling its capacity every 3 months.
Yes.
So this year's AI is 8 times more powerful than last year's. Yeah? We are 10,000s of times more powerful than in 2018 when we published our work.
Yes.
And this is what we are observing at the moment. I can tell you, we are working on a model at the moment. It's not peer-reviewed and it works, but we have created a database of 260 million data points where we have a chemical property and a result. For 4,000 of these properties, we have more than 1,000 entries. So we consider them big properties, which we can also predict, yeah, because everything where you have enough.
Yes.
And this is an interim step in this, yeah. We predict these 4,000 properties of any given chemical without sorting out the questionable stuff or whatever. These 4,000 properties with about 90% accuracy.
Yes.
So imagine even a substance which has never seen the day of light, the chemist is only considering to possibly synthesize. You press a button and you get 4,000 properties. Yeah. Whatever you could be possibly interested in. Then we are at the moment working on the integration, mapping them on adverse outcome pathways and others so that we make sense of it, yeah, because nobody can go through 4,000 properties.
Yes.
But you can, and this is getting us really into extremely good predictive values. We are evaluating this in the OnTox project of the EU at the moment for liver, kidney, and the developing brain. But it is in principle telling you whatever you're interested in.
Yes.
I did a test run with mycotoxins recently and cancer and mutagenicity data all in the 90s of predicting the test validation sets. We do safer plastics evaluation and endocrine disruptor. It is really remarkable what you can do. And we will make our database available probably in the next 2 weeks. The paper is accepted but not yet out. We are sharing through GitHub the different import functions from public databases to create such a database. We are expecting to publish our transformer model, which is predicting these 4,000 properties, beginning next year, likely, when we have done enough evaluation of the quality of these predictions.
Wow, I mean, that is amazing. And when that paper comes out, I'll be sure to put that in the show notes if that coincides with publishing this podcast. And I can see, I've seen it for myself because recently I've been looking at the mobility of chemicals in the environment. The persistence of mobility is a really hot topic. And for chemicals that are charged or ionizable, their sorption in soil is extremely complex. There's so many different potential interactions that can lead to whether a chemical will be mobile or not in soil. There's some really interesting research coming out from the University of York where they've used machine learning methods to get greater predictive accuracy than anyone's been able to do with traditional linear regression models. So yeah, it really is astounding. The use of AI in your day-to-day work, I think one thing that
I did not really answer your question. It's true.
Oh no, it's fine. It's such a big topic. But when ChatGPT came out, there was all this discussion about it was unethical. People shouldn't use AI to help them to be more productive and things. And I wonder that was an interesting take, but I get the sense that those calls have sort of died down a little bit now. But using these tools, as you say, to do research, and it's a muscle that you have to cultivate, right? Like a public speaking muscle, like a paper writing muscle, do you find?
Absolutely. I believe if we don't ride the wave, it will wash us away and we drown. So this is really important, and I'm giving lectures in Hopkins to our students about how to use these AIs, but also how to use them responsibly. Many people have looked into ChatGPT when OpenAI first announced it, in Model 3.5. It was only, I think, 10 days until 500 million users have been registered, the fastest uptake of a technology ever. And I think many people will have noticed that's something promising but gimmicky. But with these enormous developments, increase in capacity and capabilities per year, that's more or less the difference between ChatGPT 4.5 and 3.5. They really have become very strong value propositions. The most recent models, the reasoning models, they are now outperforming an academic in their field of expertise who has access to Google to answer. And they're so much faster. It is amazing what they can. It's also frightening what they can. But I believe we have to simply use them.
Yes.
And there should be no curriculum for a PhD or so where you're not leaving with basic knowledge on these things. Yeah. But you also have to know how to handle the hallucinations, how to check the plausibility, how to use the AI to solve some of its problems. Yeah.
Yes.
So, for example, I very often do the very same search twice because the hallucinations will never be the same. And I use only what shows in both of these researches. This is a very simple way of dramatically reducing these, but I also would never cite anything which I have not verified, and usually I download the papers if it's about references. But we should also be clear, AI is not just large language models. Large language models have the most beautiful user interface, and we can do a lot of things with them, but our predictive AI, for example, is only in very, very small parts using AI. And there's also no way of not using AI. If you do a Google search, since 2017, it's using their transformer architecture and is helping to find these things. You're already using it for many years. And it is just that we are now talking about it more. And I think it is really about building trust, but at the same time also to educate critical thinking. People need to understand that it is not the first shot answer they should take, that it requires really a very solid work around these things. But in general, the AI is not hallucinating more than me or my colleagues when I ask them something.
Yeah, we are imperfect too.
what do you expect from a model which was trained on the internet?
Yeah.
This cannot be more intelligent than all the median of the crap which is posted.
Yeah, exactly. Yeah, that's it. And the quality of that said crap doesn't seem to be going up. It seems to be going the other way. You alluded to the increase in the rate of scientific publication. So undoubtedly AI is going to contribute and to accelerate that. Do you have any concerns about the biases within AI, or how AI is going to interact with the reproducibility crisis, the publication bias that we have in science?
Yeah, I mean, the quality of scientific publications is for me a big concern. Yeah, as an editor, but also just conceptually, I think you said earlier that I have a chair for evidence-based toxicology, which is trying to bring the principles of evidence-based medicine to toxicology, which is really a very thorough quality control process. So I'm all in about this. And AI is helping us to solve these problems, yeah? Because we can deploy it. To give you one example, I published yesterday in Altex a draft for in vitro reporting standards, because I'm concerned about how we report on our experiments, that they are not reproducible because we are incomplete. And having checklists and these things is already an enormously important start. And we are working at the moment as a complement to this paper. We hope to finalize within the next month. We have simply created now AI-driven systems where you put in a paper or manuscript and it tells you this is how good you are compliant with the reporting standard. This takes seconds. And this makes them applicable. Yeah, this means that a journal can check, do I need to require some more information, and don't have to hope that the viewer identified these things. And it comes in seconds, and these GPTs will also become made available. We have also produced some for some of the other reporting standards, the ARRIVE guidelines for animal studies, as well as the ToxR tool, which my team did develop some long time ago, almost 17 years. But they do a very interesting job. So I'm really excited about not only producing guidance, which is dry and nobody wants to read, but a tool to implement it. And very similarly, we had an article last week in Archives of Toxicology about bias and risk of bias in toxicology. And we're discussing there, on the one hand, how AI tools can bring in more bias and accelerate it because the data translates somehow to bias in results, but that we also can deploy just to the opposite. Because if we humans can identify bias, the AI only needs to be told to do so. And can do this on scale. And so it is both. It is angel-like and devilish at the same time.
It sounds like a new arms race.
It is, but we need some literacy in this in order to be able to use these tools. And that's one goal. But we also need to systematically evaluate it and build trust. In April, I had a paper on is regulatory science ready for AI? Together with Robert Califf, at the time of writing, head of the FDA, and some colleagues. So we are really trying to describe the path forward. And this is really important. We need also new concepts for validation, for tools which are changing on a monthly basis. You cannot expect to freeze them in time and say, for the next 10 years, use this tool. It will be laughable. But you can also not repeat the validation exercise every month. Yeah, and so what can you do? And Nicole Kleinstreu and myself, Nicole was at the time still heading the US validation body. We wrote a paper in February about that we could imagine that we have an agentic system which is retraining and accompanying the validated tool to tell when it is necessary to retrain and create a new version. And then this could become a semi-automated process where people work with versions and can say, I used version 5 now. And then you can even go so far and say and alert if the re-evaluation made changes to earlier results, it would be no problem to alert the previous users. There is now a change in category for your substance you evaluated or whatever. It becomes very different, interactive. Yes. But it is something which I think is much more future-proof than freezing in time computational approach.
Yes. Well, I mean, that's it. A lot of decisions that have been made over the years that there'd be a lot of problems for to change those regulatory decisions, I guess. But yeah, I think the world you're describing just feels like a much less structured world and a much less anchored world when it comes to these things. We have to prepare ourselves to embrace much greater complexity and fluidity in how we're approaching things.
Sure, but we also have the tools to help us with this.
Mm.
I mean, when I was working as a programmer, we needed databases with good annotations, and now the new systems often work with structuring on the flight. Yeah, they are ordering the data while they import them, and can work with completely unstructured information. Yeah, that's how you get a very meaningful excerpt from a YouTube video or whatever within minutes.
Yeah.
It is really remarkable how AI can bring structure into things which are not structured yet and help us to digest these things.
Yes, and connect things, like you said, connect disparate things to gain new insights that are totally beyond our capability. I think that brings us nicely on to the point that I originally reached out to you about, which was the article in Nature Medicine, how AI can deliver the Human Exposome Project. Would you mind just saying what the Human Exposome Project is?
Yeah. In 2005, Chris Wilde, who was at the time heading IARC, the cancer agency in Lyon, came up with the crazy idea to say we should study the human exposome, the totality of things which are impacting on humans. At the time, it sounded terribly naive that anybody could attempt something like this. Yeah, but I come to the conviction more and more over the years that we actually need a complement to the human genome. I said already, 5% of diseases are genetic, 40% have a component which is genetic, but 80-90% of chronic diseases have an exposure component. Yeah, and this is these are the big ones, the expensive ones. Yeah, so we really would benefit from understanding more here. And from the strategic point of view of changing toxicology, moving away from animal testing, also, I think it might offer to us the opportunity to move toxicology from the art of poisoning rats, or let's say for CETAC, the art of poisoning zebrafish, to something which is more the exposure side of disease. Yeah. And this is the idea of the human exposome. So that you are using more or less the rich information you can nowadays get from omics studies in biofluids, monitoring data on the diverse chemicals we can identify in samples, and that you form from this an exposure hypothesis. It will say, okay, I can explain certain patterns both in the chemicals I find, the metabolites I find, but also then the metabolomic changes, the transcript changes in these people, that I can come up with a hypothesis which is saying it could be that there is a pattern, that a certain class of chemicals or a certain individual chemical could be associated here with some changes which are meaningful and should be followed. And then you would enter into a phase of trying to verify or falsify these possible associations experimentally by using human relevant systems. Here we come back to the microphysiological systems.
Yes.
And the hope would be, by building more and more of these associations, learning from these data, we build up something which I used to call a human toxome at some point, but we are now thinking more and more of digital twins, of systems which are reflecting biology and the perturbation of biology through chemicals. That's the big vision. And we were twice very lucky in the last year. The first time we were very lucky that I convinced our major philanthropic donor to make $17 million available for the next 7 years to prepare for such a human exposome project. And then my deputy, Fenacier, did succeed in getting an idea competition through for Johns Hopkins' new fantastic meeting space in Washington. So this is really a beautiful place, up to 360 people can host, and directly at the Capitol, you can talk to policymakers quite easily. Yeah? And then we said, why don't we change gears, and instead of working now 7 years towards the exposome, why don't we invite for this venue the leading experts in exposomics and the civil society, the people you need to convince that it needs a human exposome, policymakers. And we found, despite the fact that we only had a year time, we had no major sponsors, we did no industry exhibition and nothing, very minor sponsorships only. We brought together 300 people and were endorsed by 4 NIH institutes, 2 of them in the organizing committee. We had the WHO represented with Sir Jeremy Farrar, at the time, chief scientist of the WHO, and a day later he was promoted to Deputy Director General. We had Rémi Quirion, the president of the International Network of National Government Science Advisors, Peter Gluckman from the International Council of Sciences, the umbrella organization for 250 scientific organizations in the world, or the CEO of the African Academy of Sciences. So really top people. Even the new head of NIH requested an invite. In the last moment had declined, did not come, but just that our event, he requested to come and join us for a while. This was, I think, big success.
Yes.
And it really showed a momentum that people felt, yes, we can, yes, we should go, and we are starting now working groups, and not just about the science, but it's a mapping of projects, but it's also a lot about what would be the governance of such a project.
Yes.
What could be funding streams? How can we quality assure things, best practices and others? Yes. It's an exciting time which really puts on a completely different scale these transformative technologies, but it's just the same technologies which we are using here.
Yes, I mean, it does sound totally game-changing, to be honest, the whole exposome concept. And as you said, AI has really kind of made this a possibility, because we're talking about all kinds of different data that we might feed into this kind of a framework, from exposures, pathogens, physical exposures, but also genetics, lifestyle. I mean, it's so diverse.
Yeah, but AI shows us every day how it is capable of integrating more of this. I mean, it has also its problems, because we see how much energy it takes to crunch so many numbers. But, we are also developing the solutions. Quantum computing is promising to be 1,000 times more energy efficient. So if this is coming, if we have a problem, we often can also solve it. I don't say we should blindly follow the science, but on the contrary, my learning with validation and all of this tells me, yes, but be a self-critical bystander also. Just observe what is happening. Does it make sense? And where do we need guardrails to not go astray?
Yes, the sense I get is there's a huge amount of optimism from yourself and excitement about all these technologies, but like you say, you're also conscious of the power of these tools and what could go wrong.
Yeah, and there's no way of not trying it. Yeah,
no.
Failure is an option. Not trying it is not.
We just have to hope that we have responsible people in the right places, doing responsible things as we go, which we're all accountable for. Everybody who's involved, I suppose.
This is why we need also this division of power. Yeah, we need some people who develop these things. We need separate people who are validating them, and then we need another group of people who take decisions based on this too, because you need all three. Yeah.
One of the things that really interests me about the exposome is because in my field of environmental chemistry and environmental persistence of chemicals, this issue has really kind of grown hugely in recent years. Everybody's really interested in whether chemicals will degrade in the environment. I think this has largely been triggered by issues like plastic pollution and also PFAS, both regarded as not degrading in the environment. And I think it's also linked to the sustainability movement and the drive towards a circular economy, and whether emissions of non-degradable chemicals into the environment can be considered a sustainable activity. But I think something that strikes me is that I really get the sense that the public doesn't fully comprehend the nature of the society that we've built and the nature of their exposure to chemicals in their day-to-day life. I feel that they can get on board with the idea of, if they work in an occupational setting, there might be exposure to some chemicals, or if they use a product in the home, they might be exposed to chemicals. But especially when it comes to environmental exposure, and how a chemical could make its way through the environment back to people. I think that's something that is totally alien to a lot of people. And as a result of that, we see alarming headlines all the time about certain chemicals being found in food or people's blood or things like that. And my thoughts on exposome is that it's also an opportunity to bring the public to a greater awareness about the nature of the society that we've built, and that hopefully that will help us to have better policy decisions as well.
Yeah. No, I agree. I mean, the number of chemicals which are around us and are being used is incredibly high. Yeah. I mean, I can start with there's about 1,000 chemicals, industrial chemicals making it to the market in the US per year. 1,000. No way of assessing them properly before they come. A study from 2019 has shown that there were 350,000 chemicals registered for marketing in the 19 most developed countries. 350,000. Yeah. Nobody will ever be able to address them comprehensively except with tools which are purely computational to identify problems. And I think this is really key. I don't think that chemicals are bad, but we have produced enormous chemophobia because of the lack of knowledge and some cases which have been so much discussed. And I think we need to change this narrative because, I mean, we have tripled life expectancy since these chemicals came into our life. Yeah,
Indeed.
So they cannot be so terribly bad. And there's not many diseases which are really increasing if you correct for age. So it is really important to understand that we probably need only a few culprits, certain things which we need to identify. And again, this is probably happening through screening, yeah, not through definitive studies and testing every chemical on everything. Yeah. So there's a lot to be done, and it is quite fascinating that the movement of green chemistry to make things more sustainable has two of its 9 principles, which are toxicological principles. It is benign by design and early identification of toxic liabilities.
Yes.
And it is the very same technologies which we need, as we discussed before. It is computational, that the chemist can design safer drugs or safer chemicals. It is the testing, early testing, which is only possible with the tiny amounts of an in vitro system to make use of them. So I was, interestingly, just the next week after our Moonshot Forum, Exposome Moonshot Forum in Washington, I was in Stockholm for a Nobel conference for the future of green chemistry. There,
Yes.
under the lead of Paul Anastas, one of the fathers of green chemistry,
Yes.
30 people, me involved, had developed a declaration for the future of chemistry, which is trying to promote exactly the same idea. We need change, and we need to build this change on these transformative technologies.
Yes. Yes, I'm glad you brought up that declaration, because that is something really important that's going on right now, and I would encourage everybody to read that declaration, and if they choose to also sign it. I've read it several times, and I think it does lay out some very good ideals and principles for how we can move towards a more sustainable society. But I think as well, one thing that I'm sort of starting to see is that we almost need a deeper philosophical discussion about the environment. And because I think underpinning a lot of the discourse around chemicals in the environment are the fact that there are two worldviews. There's a human-centered worldview that sort of accepts and aims to manage damage to the environment through human progress. And there's another worldview of people who simply want to preserve the environment for its own sake and leave no mark on it. And it seems that until we revisit that, we're going to continue to have fundamental disagreements about how we can manage chemicals.
Yeah. No, I agree. And I'm always attracted by these type of big discussions. Yeah. I think the One Health discussion is an interesting one. The planetary health discussion, this is what you're alluding to. I think both are very important, and we need a good understanding and modeling also of these connections for doing the effective things.
Yes.
It was quite fascinating, and at this conference on green chemistry and the Nobel Foundation, when people were showing that the ecological footprint of a paper bag is much bigger than of a plastic bag. So we are sometimes doing not the things which are intuitively not the correct things without being able to judge all of these arguments. In toxicology also, we do sometimes really some crazy discussions, and take bisphenol A. That's a discussion which has led to 12,000 scientific papers. And we're still arguing whether some extreme exposures of babies could have an endocrine disrupting effect. It is certainly not the biggest problem in the world, because we are swimming in endocrine active substances in all of these phases, and it is, if at all, a borderline exposure. But we have tremendous scientific effort without coming to a clear result. Which is crazy, with respect to resources, And you can also say, let's be precautionary and simply say, we use something else. Go back to glass bottles. I don't know. There's advantages and disadvantages to all of this, but I think a more comprehensive view on things is warranted, also to avoid regrettable substitutions, or to just not. There's always this joke that a physician is dealing with a cancer patient and is so much worried about some little detail on the body, which he's specialized in and working on, and this is exactly what is happening. We have major problems and we need to find solutions. And I don't like that we have not found a way of making the human exposome a project which also encompasses the ecological aspects, because I think the same tools could be applied.
Yes, definitely.
But profiling it as a complement to the human genome, it made sense to make it human again.
Yes.
While we are falling short in understanding and including some of these important aspects of environmental health and its impact.
Yes. No, I really appreciate those reflections, Thomas. And this has been absolutely fascinating discussion. I knew it would be. So I just want to thank you for your time today. For those listening, Thomas is on his holidays at the moment, so he took the time out of his vacation to speak on the podcast. So thank you very much for that. And thank you to everyone who's been listening. Your time's really precious, and so I'm really grateful that you've spent it with us. I hope you've enjoyed this discussion. If you have, please tell your friends and colleagues about it. Spread the word about the Chemical Journeys podcast, and I hope to have many more conversations like this to follow. Thank you very much, Thomas.
Thanks for having me.